you first launch the VIROME web application, you're brought to the home page. On the left-hand side of the home page is some text explaining the VIROME ORF and sequence categories. These are the categories that every sequence and every ORF is placed into based on the bioinformatic analysis within VIROME. Below that is a table of the publicly available viral metagenomes and data within VIROME. And then above that is a bar chart showing the 6 different environments that the VIROME data is placed into. Extreme environments, organismal substrate environments, which would be host-associated environments, sediment environments, soil, solid substrate, or water environments. You click on one of these bars, it then breaks the data down into the 9 different functions. Clicking on one of these bars will go through to the Browse page, which can also be accessed through the Browse button. You'll notice here that all we can see are the public libraries available in VIROME, and again, they're organized into these 6 different environments. But if we log in, then the private libraries that are available to us are shown here in the same interface. On the Browse page, there are 4 columns. The first column gives some basic information about the library. The second column shows a pie chart of the ORFs and sequences organized into the VIROME categories. The third pie chart shows the ORF types, either lacking ends or complete. And then the fourth column shows a pie chart of the taxonomic origin of blast hits against VIROME ORFs. All of these charts can be downloaded, and the data can be downloaded as tab-delimited files. It's also possible to click on any of the pie charts, which will take you to the search page. We'll name the search. In this case, I'm going to name it functional protein. And submit that search. When the search results are ready, a dialog box will pop up. You can click View Now, and this will populate the search results pane with all of the information about functional hits, meaning hits against the UNIREF 100 database. All of these columns can be sorted according to their sequence description or the organism, and this data can be downloaded directly by clicking the download search sequences. We have 4 different types of downloads that can be done here. Comma separated values that you see here in the search pane, a FASTA file of the peptides, a FASTA file of the nucleotide ORFs, or a FASTA file of the reads. Here in the sequence details page, I've actually uploaded a library of all the functional proteins against some contigs to show you that we can display long reads of data in the VIROME sequence details. So here you'll notice I've sorted the sequence name column, and I see this particular contig here, 1021, which actually has 9 ORFs on it. I can tell by this last underscore. So we'll go ahead and click on that particular ORF, which brings up its sequence details page. The sequence details page shows all of the information that we gained about this ORF from the blast analysis. This heat diagram here, we can click on one of the squares in the heat diagram, and this brings up the blast results for that particular ORF against the UNIREF database in this case, or against the Metagenomes Online database in the other case. Then I've got a list of the data organized by the actual databases. So UNIREF, SEED, KEG. These accession numbers are hot linked to the actual UNIREF database, so I can get to the UNIREF sequence entry for that particular ORF. I can do the same thing for SEED or KEG. Then, of course, this was the ORF, which came from a read. In this case, the read was a longer contig. This map shows all the position of the ORFs along the contig. And then down below is a summary of the hits of the ORFs against various databases. So here we have Uniref, Seed, KEGG, COG, and so on. These are also hot linked to their ORF, so it's possible to click on that and then go straight to that particular ORF sequence details page. So this is really the finest scale of detail in the VIROME web application. The search page is fully interactive, so it's possible to search a library according to descriptive terms. Even if you know the sequence ID, it's possible to search by that, to restrict your results by a certain BLAST E-score, to restrict your search to particular VIROME categories, one of the 9 sequence categories, the type of ORF, whether it was complete or missing ends, even the taxonomy of the taxonomic origin of hits is all searchable through this interactive search pane. And when a search is run, it populates this search pane here where it shows the search results. And then as shown before, you can download those sequences as a comma-separated values file or as FASTA files of peptides, ORFs, or reads. Each search that a user runs is stored in the VIROME web application. So it's possible to go back to previous searches. So you click this bookmark button on the menu bar, and you can see a list of the search date, the number of records in that search, the name that I gave to that search, and then information about the library and so forth. So I can click on this previous search, and the results of that search will populate this search pane. In VIROME, it's also possible to look at entire libraries of data through our statistics summary page. So I can navigate back to the browse page and choose a library, click on that link for the library, which will take me to the statistics page where where a lot of data is summarized about that library. So here we have text information, summarized information about the library, its location on a Google map, the length histogram of sequence read lengths, ORF lengths, and the GC composition on this pane. Then the VIROME categories summary for this particular library is shown here. All of these charts can be downloaded, or the data behind these charts can be downloaded. I can view information about the tRNAs that were identified within this sequence library. I can download this table of information about those tRNA hits. Among the possible functional proteins, these are the organization of the hits against the various annotated databases. It's possible to click on one of these and it will take me to actually the seed page, and I can see all of the hits in my library against the seed database organized by the seed categories. Each of these will navigate to the next lower level. And then if I double click on any of these bars, again I can run, it will run a search. And then I can view those search results here. So these, that's a way of, again, organizing your data and binning your data. Can do the same thing for any of the data. So for instance, if I go to taxonomy, can navigate through the taxonomic levels. Here we start at the highest level. At the kingdom level, can navigate down by clicking through each taxonomic level. And I'm going to click all the way through all the way through to cyphoviruses, and then run a search. And then get those search results up in the search window here. Finally, if you are interested in learning more about the Virome bioinformatic pipeline, you can click on the About button here on the menu bar, and we have an interactive conceptual diagram that illustrates various components of the Virome pipeline and how sequences are processed. Thank you for your interest in using Virome. Virome is supported by grants from the Gordon and Betty Moore Foundation, the National Science Foundation, and the U.S. Department of Agriculture.